All right, so in this section we're going to look at the membrane in more detail in there. One of the key things about our membranes is they have selective permeability. Some things can get in, some things can get out, and it gets to choose those things in there. Your cell membrane is going to be a fluid mosaic model of this phospholipid bilayer. So remember, your phospholipids, we tend to draw them with the phosphate head, two fatty acid tails. With the bilayer, what it means is we've got two layers that are turned towards each other in there, so that we have fatty acid tails in the middle and we have phosphate on the outside and the inside. What this is going to create is an amphibolic type of structure in here. On the outside and the inside, this is going to like water. However, in the center area here, it does not like water. This is going to be good for having a watery substance inside your cell, watery substance outside your cell, but they don't want to go through this no man's land in the middle to mix together. It will help keep them separate in there. With the fluid mosaic model, what that means is you've got all of these phospholipids and proteins embedded in between them, and all of those things can slide around amongst each other. The easiest way I can think of to picture this is if you take that triangle shaped thing that you put pool balls in on a table, and you say you get most of the balls in there, but they're not all in there. You can scoot them around and rearrange them in there. You don't actually take them out, but they can move around, and you can take your black ball and you can put it in the corner, or it can slide around and move around amongst the other balls. Your cell membrane does that as well. So this can slide around and it can move not only side to side between these, but out in other directions. So it moves all around the outside like that and can rearrange. So these membrane models have not been around all that long. In 1915, membranes from red blood cells were analyzed, and they found out they were made up of lipids and proteins. In the 1960s, there was more of the sandwich theory that was made in there by Dawson and Daniel, where you have the phospholipid bilayer between two layers of proteins in there, and that model was widely accepted for a long time. In 1972, you have the Singer and Nicholson proposed membrane in there that is going to have the proteins individually inserted in the phospholipid bilayer. So, you know, I mean, that's fairly close to when I was born. That's not that long ago, right? Okay, yeah, when we look at it and we say, oh my God, it's over 40 years ago, it sounds like a long time, but that's going to make me old. Yeah. So the fluidity of the membranes, being that these adjacent phospholipids can switch positions rapidly, they can do it 10 to the 7th times per second. What that means is they can do it this with 7 zeros. They can move around that many times in a second. That's a lot of movement in there. When you put the proteins in there, they're going to be larger, so they're not going to move around quite as quickly, but they're still going to move around at a good pace in there. A lot of times the proteins are going to drift in a highly directed manner in there, so they're going to go and move around for a specific reason in there. So as the temperature drops, the fluidity of the membrane is going to decrease. So just like if you were to take a cube of butter, when you put it in the refrigerator, if you want to mix up that butter, it's not going to be very easy. It's really hard. However, if you take that butter out and you leave it outside on a hot summer day, real easy to mix up that butter in there. It becomes much more fluid. Same thing with your membranes in there. So the idea that your body would actually kind of start to stiffen up in the cold makes a lot of sense if things start to get colder, the tissue will get firmer because of the membranes changing in there. Cholesterol is important because it's going to make the membrane less fluid at body temperature in there, and it just helps to solidify it and act as a temperature buffer. Because it's not going to be as sensitive to the changes, it's going to keep you from getting too stiff in the cold so that you can go outside and your skin doesn't necessarily just crack and become brittle, but it also keeps you from melting if you go into the desert in the hot summer. So these membranes that are embedded in the these proteins embedded in your membrane, we have over 50 different kinds of membranes that we've identified so far. And that's in the plasma membrane of a red blood cell. So there are very likely thousands of these. Most of them we don't know in there. And when we look at their functions in there, we've tried to kind of group them into what they do and categorize them, rather than saying this is protein A, it does this is protein B, it does this. Rather looking at, say, this group has this type of behavior, this group does another type of behavior. So your integral proteins are going to be the ones that penetrate to the hydrophobic core of the lipid bilayer. Your transmembrane proteins are going to span the entire membrane. So which one of those do you think this is? That would be a transmembrane in there. An integral just stabs into it. Doesn't have to go all the way across in there. So they can do either one in there. Peripheral proteins are not embedded. These are actually going to be appendages that are bound to the outside surface. So that could be another protein that's stuck out here on there. And then the integrins are integral proteins that attach to fibers of the extracellular matrix. So then you could have the integrins attaching on the outside, the little fibers in there. So we have 6 major functions that are performed by these proteins in the plasma membrane, transporting things. It's a good way to get things in and out of the cell. Enzymatic activity, so they're going to control enzymes, chemical reactions in the cell. Signal transduction, if you want to send a message to another cell, you're in distress, you can send it out through these. In there, cell-to-cell recognition. So this way you can tell when you're with your own kind in there. When you make new cells, they don't just randomly end up somewhere in your body. When you make new liver cells, they're made in the liver and they stay with the liver. They don't end up going to your lungs in there. Intercellular joining, so that you can attach to your neighbors. It helps to also attach your cytoskeleton and extracellular matrix. So an example of one of these being able to do some cell signaling in there would be one that you have that is going to signal for fat. So if you've ever wondered why when I gain weight does it always seem to want to go to one particular place in your body? Because some places in your body you have a marker on there that is going to call and signal to the fat, come live here. So far we can't change where those markers are. So our membrane carbohydrates, these are going to be sugars involved in cell-to-cell recognition. Glycolipid in there, so the name tells you glyco for a carbohydrate, lipid for the fat in there. It's going to be a short membrane carbohydrate that is covalently bonded to a lipid. Glycoprotein is going to be your lipids covalently bonded to a protein in there. And you've got your glyco for your carbohydrate portion in there. There is a great deal of variation in these carbohydrates that are on the outside of your cells like that. It's been said that because of all of the variation in these markers and everything it does, the cell membrane is probably almost as important as the DNA in directing what the cell does in there. So this picture here is just illustrating again that some of these don't go all the way across, some of them do go all the way across, some are attached on the outside, and that you've got the other proteins attached on there. So to synthesize and your sidedness of membranes, they have a directional orientation. They can't go just either way. They aren't the kind of thing you flip-flop on there. They're going to be synthesized in the endoplasmic reticulum, and then they get converted to some glycoproteins. After they're made in the endoplasmic reticulum, you send them to the Golgi apparatus in there to make some glycolipids. Your transmembrane proteins, your membrane glycolipids, and secretory proteins are going to be transported to vesicles, where they'll be transported to the membrane in there. So just thinking back to earlier, which face are they going to go in when they get to the Golgi apparatus? Which side do they go in? Cis side, and they come out on the trans side. So then they will fuse with the membrane, and it releases the secretory proteins and the glycoproteins to get positioned on the outside. So if you've got this fluid mosaic model on there, basically you just kind of can spew those new parts in there, just like throwing a few more of those balls in your pool triangle that you're setting up. So the structure of the membrane is what is going to result in the selective permeability in there. You've got your nonpolar hydrocarbons, like carbon dioxide and oxygen, that are going to be able to cross that hydrophobic core easily. So your hydrocarbons in there are things that are like fats. They're going to be able to get through this. This area isn't that strong to keep them out. So when they get into this middle area, they're not bothered by that. It's like dissolves like likes to be with like in there. So you can have the fatty things come through this. Your steroid hormones, if somebody takes a steroid, what is one of the things we see as a problem with steroid drugs? They tend to have a lot of side effects in there. So we'll just say you want to abuse some anabolic steroids and you're a female, and they're just because these effects are going to be greater on you. You may inject them into your muscle, but it doesn't mean they're going to stay there. They are able to diffuse and move around quickly. So they're going to diffuse to your skin cells and get in there and have an effect of making the skin thicker and coarser, more masculine. It's also going to trigger more hair growth in there. It will diffuse up to some of your cartilage. It may give you an Adam's apple in time. Ooh, just what every girl wants. And also, it will go to other places in your body and cause more masculinization in there. So the downside to steroid hormones is we may want them to go into the body for a specific effect when we give them to a person, but they tend to diffuse and go all over. Lots of side effects because they can cross that passage in there. So oxygen and carbon dioxide are really small, which allows them to go in and out of there real easily in there. So your transport proteins, these are going to be permeable to specific ions and polar molecules. So this is going to allow some of the hydrophilic substances to avoid contact with the lipid bilayer. So it basically can make a channel. So if you want to go through here but you don't want to touch this stuff, you just go through the channel here. The aquaporins are going to be channel proteins that are specific for water molecules in there. You've got some carrier proteins in there that are going to hold on to a chemical passenger. So this is kind of like you get to freely swim across the channel. With your carrier ones, you get like a little ferry ride across the channel where you're going to go on a carrier protein in there. And then with this picture here, this is showing some of the ways that things can go across in there. With transport, this one is showing that it's going to use ATP in there to bring something across. This one showing enzymes coming in, and it's going to change the shape of it bringing it across where you have a signal coming in and meeting up and passing the signal along, attaching to the cytoplasm, joining to the neighbor membranes, or making glycoproteins so you can recognize your cells. These glycoproteins are actually really important for recognizing cells. If you've ever wondered why you can have a cold or a flu, and if you have pets, you know they do kind of disgusting things like eat Kleenex and things like that. Yet your dog doesn't get the flu from eating the Kleenex in there. Is certain viruses need certain ones of these glycoproteins in order to infect the cell. If this isn't there, they don't get to infect it. So it doesn't matter if you don't have this glycoprotein that the virus needs, bring it on, you're not going to touch me. So that's why your pets can be around you during those things. However, I certainly wouldn't go eat Kleenex just because you feel like you couldn't be infected by something. So passive transport, this is going to be diffusion of a substance across a membrane. No energy is required. These are substances that are just able to cross anywhere on the membrane in there. It's usually going to be that they're going to try and move so that you have an equal concentration of the substance on both sides. The concentration gradient is going to control it. So if you have a whole bunch of dye molecules on one side and just water on the other side, they want to go across this membrane, and more of them are going to end up going over to this side here until you reach equilibrium where they're the same on both sides. So in one of the labs, what you'll be doing is we have some dialysis tubing, and you put in a dye that will stain starch, and you put the starch in there and wait for the two to mix. And this is just showing it happening with more than one substance in there. So when we look at that specifically for water, we refer to it as osmosis in there. And it's going to move from a region of lower solute concentration to a region of higher solute concentration. Basically, if there is something that's more concentrated, it always tries to dilute it. And that's not just a cell or a body doing it. That's just a principle of nature in water with chemistry in there. So if you don't have cell walls, you're not going to be as tolerant to excessive water losses or gains in there. So the tonicity is the ability of a solution to cause a cell to gain or lose water in there. If something is isotonic, iso means the same, it's going to be the same concentration on both sides. So things will move back and cross, but they do it at the same rate, so there's no net change on either side. A hypertonic solution will lose water to its environment, shrivels up and dies. Hypotonic solution, water enters the cell faster than it can leave, and the cells will burst. So I always forget to water plants. What kind of situation do I put my plants in? Yes, my house is a giant hypertonic environment for houseplants. You will die. So osmoregulation is just going to be the control of water balance in there. When a cell has a cell wall, we've got turgor in there, and that means the cell is firm due to swelling, and the inelastic wall is going to put pressure back on it to oppose further water intake. That's a healthy state for most plant cells to be in. So at one point, I had been given, I think they're like a Gerber daisy or something like that. Some daisy they're actually kind of cool. They have a big flower, daisy style flower on them. And I was really trying hard to keep the thing alive. And I go out there and it's just bent and hanging down like this. And I think, well, okay, maybe I can bring it back. And this plant was actually pretty cool because I put a whole bunch of water in it. And like an hour later, it stood back up in there. So I restored its turgor in there. Plasmolysis is going to be when it's in a hypertonic environment and it loses its water and wilts. So with facilitated diffusion in here, this is passive transport aided by proteins. So certain hydrophilic solutes are able to cross passively with the help of a transport protein. These are the ones that need to take a ferry across, or they need to go over a specific bridge to get there. So water is small enough to cross the lipid bilayer, but it's very slow. But if you put it someplace where it's got an aquaporin, it can cross it much more quickly in there. So here it's showing you've got the coming in, the carrier protein, moving it across in there. So you can have some gated channels in there that are going to be ion channels that open and close in response to stimulus. So for example, in a muscle, you're going to have a gated channel that's going to allow sodium to go. If you stimulate the muscle, it will open the gate for the sodium to come out. If you don't stimulate the muscle, that won't happen. Glucose transporters in there, these will undergo a subtle shape change to translocate and bind things across a membrane in there. Then with active transport, these are going to use energy to move the solutes against their concentration gradient. Because you want to go the wrong way, you're going to have to pay to do that. And so they're usually going to pay with ATP. So an example of doing this is a sodium-potassium pump in there that's going to move sodium and potassium. This is actually huge in your cells. This is how you spend about 1/3 of your calories every day, just your regular calorie intake without exercise, is moving things for your sodium-potassium pumps. What it's going to do is it's going to move 3 sodiums out this direction in an area that already has a lot of sodium. It's trying to build up that sodium concentration. Then it's going to move 2 potassiums in the opposite direction. So what you end up with is lots of sodium on one side, lots of potassium on the other side. So what's going to happen if you open up a channel in there? What do these naturally want to do? They want to naturally mix together and come to the other side. So what you're doing when you do those sodium-potassium pumps is you're essentially like charging up your battery in there so that when you want to send a message down a nerve cell, you can open up the channels in here and these move together. But you're constantly separating it and recharging that battery to be able to do that. So when it does that, it actually will create some membrane potential or voltage in there. It's just like you have voltage from a battery. When you look at a battery, you've got a positive and a negative end of it. You've essentially created a positive and a negative here. So we say that you've got membrane potential here, just like this battery has potential to do work. This has potential to do work in order to get to let those charges come together or those ions come together. So this battery will do work for you if you stick it in something where you connect these two ends and allow the positives and negatives to come together. This will do work for you, like send a nerve message, if you allow open gates and allow these to come together. So it's an electrochemical gradient that's going to drive that in there, and we refer to it as an electrogenic pump in the animal cells in there. We will also create this with a proton pump in here, where you can have your protons on one side, your electrons on another side. Because these are positive and negative and want to come together, if we create a spot where we're going to let them come together in there, they will do work for us in exchange for getting to come together. That's going to be what you'll do in making ATP. So sometimes we're going to have co-transport in there. It's going to be an ATP powered pump in there where you're going to have one substance go one direction, another substance goes another direction in there. So this is just showing it in a plant cell that you'll have sucrose. This one, they're actually going to go together. Sucrose will travel with the protons incoming across. Oh, yeah, we're almost done. So bulk transport. Sometimes we want to move things that are bigger across the plasma membrane, and they're not going to be able to just go through a transport protein in there. So this is when we do endocytosis and exocytosis. For exocytosis, this is where you're going to excrete a substance in there, and what it's going to do is it's going to come in a vesicle up to the membrane. That vesicle is going to fuse with the membrane in there, and when it does that, it opens up and allows the contents to go out. So it's kind of like if you're chewing gum and you try and stick your tongue through the gum in there, you eventually burst a hole in it, and it allows your tongue to go through and spew out whatever's in there. So this is how you're going to do things with a lot of the secretory cells. This is how insulin will be released. Your neurotransmitters and your neurons in there are that way. With endocytosis, it's going to be the opposite. This is how you're going to take something in. So with phagocytosis, it means cell eating in there. The cell is going to engulf the particle by wrapping itself around. Cell eating implies that it is a solid particle in there. Penocytosis, cell drinking, that implies that it's a liquid. Usually that designation is only used in cell textbooks in there. Most of the time you'll see endocytosis or phagocytosis used. With receptor-mediated endocytosis, this is going to allow you to acquire bulk amounts of a substance, even when they may not be concentrated in there. And so what it does is it allows you to build them up. It's showing here where you've got these membrane-coated pits in there that will have these vesicles containing ligands in there, will ingest that, and then it allows it to go inside. And those receptors can be recycled again. So that is the end of the cells. Is there any questions on that?